Hippocampus and its role in encoding time and experiences. Henry Molaison was born on February 26, 1926, in Hartford, Connecticut. He had a typical childhood, but by the time he was 10 years old, he began to suffer from severe epileptic seizures. He was hit by a bicycle when he was 7 and suffered a head injury which is believed to have contributed to the onset of his severe epilepsy. As he grew older, the seizures became more frequent and debilitating, making it nearly impossible for him to lead a normal life. By the age of 27, his condition had worsened to the point where he could no longer work or even perform daily activities without the threat of sudden seizure. Desperate for relief, he sought the help of Dr. William Scoville, a neurosurgeon who suggested an experimental brain surgery called a bilateral medial temporal lobectomy. At the time, the procedure seemed to offer a chance to alleviate the severe epilepsy that had taken over Henry's life. During this procedure, Dr. Scoville removed a part of the brain that is known as the hippocampus. The hippocampus is a small, curved structure deep in the brain, and because of Henry's case, we now know that this structure is crucial for the formation of new memories. And we know this because while after the surgery, Henry's seizures diminished, he was left with severe anterograde amnesia, the inability to form new long-term memories. His old memories from before the surgery were largely intact, but every new experience slipped away like water through a sieve. After the surgery, Henry's life became a series of moments that he could not string together. He lived perpetually in the present, unable to remember what had just happened a few minutes before. He could hold a conversation, but if he left the room and returned, he would greet people as though he had never seen them before. His sense of time was frozen in 1953. Despite his condition, Henry could still learn new skills, a phenomenon known as procedural memory. For example, he could trace a star while looking at its reflection in a mirror, a task that requires hand-eye coordination and practice. Yet each time he was introduced to the task, he would insist that he had never seen it before. This suggested that the parts of the brain that are responsible for procedural memory, which are now known to be the cerebellum and basal ganglia, are still functional. Brenda Milner, a neuropsychologist, began working with Henry soon after his surgery and documented his condition extensively. Through her studies, Milner and other researchers discovered that memory is not a single process, but consists of multiple systems. They distinguished between explicit or declarative memory, which involves consciously remembering facts and events, and implicit or non-declarative memory, which involves skills and habits. However, the fact that procedural memory remained intact showed that while the hippocampus is critical for forming new explicit memories, other types of memories can remain intact even when it is damaged. Through Henry's case, the function of the hippocampus acting as a processing center that helps convert short-term memories into long-term memories is now better understood. But even now, 16 years after the death of Henry, scientists are still working on how memory forms in the brain and how we remember the sequence of events.
The hippocampus, along with surrounding areas such as the entorhinal cortex, play key roles in spatial memory, and two types of specialized neurons, place cells and grid cells, are central to this process. Place and grid cells enable the brain to represent space and navigate through environments by creating an internal cognitive map. These cells are fundamental for forming spatial memories such as remembering the layout of a room, the location of an object, or how to navigate through a city. The hippocampus and entorhinal cortex collaborate to encode these spatial representations and store them in long-term memory. The replay of place cell sequences during sleep further strengthens these memories, allowing them to be recalled and used in future navigation tasks. When exposed to a novel environment, the grid cells provide the spatial metric for exploration, and the place cells form new place fields that correspond to specific locations. This process allows the animal to learn the layout of the new environment and remember it for future use. Try to explain the last exciting football match that you have watched to one of your friends. Which team scored first? Which one performed better in third quarter? Did your team have a comeback in the last seconds? How does your brain keep the track of time? How do we remember what happened first and what next? A groundbreaking study conducted by UCLA Health has shed light on a fundamental mystery in neuroscience: how the human brain perceives and understands the passage of time and experiences. The research demonstrates how certain neurons in the hippocampus and entorhinal cortex encode time and experiences, playing a crucial role in memory formation and predicting future events. Imagine you accidentally push on a door that says pull. The next time you approach that same door, you might remember your previous experience and avoid making the same mistake. This learning can also apply to other doors you come across, as the experience is similar and aligned with the patterns you previously learned. These research findings offer the first concrete evidence of how certain brain cells combine what and when information to capture and preserve representation of experiences over time. To investigate how the brain processes events over time, the UCLA study enlisted 17 participants with intractable epilepsy who had previously received depth electrodes implanted in their brains as part of their clinical treatment. The researchers monitored the participants' neural activity while they engaged in complex procedures involving behavioral tasks, pattern recognition, and image sequencing. Experimental paradigm capitalized on the fact that the human medial temporal lobe contains neurons that respond selectively to particular people. Neuronal activity was recorded from multiple brain regions, including the hippocampal, entorhinal system, and amygdala. By watching the neuronal activity, scientists could observe which neurons become activate in response to looking at images.
The participants then completed a screening session in which they were shown around 120 images of people, animals, objects, and landmarks repeatedly on a computer over a period of about 40 minutes. 6 images of people associated with robust and preferential responses of single neurons in the medial temporal lobe of each participant were selected for the main experiment. Afterward, the participants took part in a three-phase experiment during which they performed behavioral tasks in response to images that were randomly displayed at different positions on a pyramid-shaped graph. Each participant was assigned 6 images for this phase. In the first phase, the images were shown in a pseudo-random order: 60 direct and 60 indirect graph transitions. In the second phase, the sequence of images was determined by their positions on the pyramid graph. The final phase repeated the pseudo-random order from the first phase. While viewing these images, participants were asked different questions about the images, such as what is the gender of the person shown, or determining if an image was mirrored compared to its appearance in the previous phase. A fascinating finding emerged from the concept of successor representation. It models how often a neuron anticipates encountering a future state, such as an image after being in a current state. For instance, if one image frequently follows another in a sequence, the neural activity associated with the first image will start to reflect the likelihood of encountering the next image. This approach combines information about the sequence's structure and helps predict future events. Over time, the neuronal responses adapt to encode not only the specific image being shown, but also the relationships between that image and others that are directly or indirectly connected within the pyramid structure. This finding suggests that the brain uses predictive coding to learn about the temporal structure of experiences. This allows it to anticipate future events based on past sequences, which is crucial for memory and decision making. Another interesting finding is that neurons are able to replay their experiences, which allows them to reinforce what they learned throughout the day during breaks. Neuronal replay refers to a phenomenon where sequences of neural activity representing a previously experienced sequence of events are spontaneously reactivated during periods of rest or inactivity. This mechanism is known from studies in animals, where play cells replay sequences of locations during rest after navigating through a maze. Fried and his team find that neurons fire in a specific order that mirrors the temporal sequence experienced during the exposure phases. The analysis focuses on three-node trajectories in the pyramid graph. These consist of three images or nodes that participants encountered in a specific order during the exposure phases. For each trajectory, the researchers identify triplets of neurons: a seed neuron, green, that responds preferentially to the first image; a direct neuron, magenta, that responds to an image directly linked to the seed image on the pyramid graph; an indirect neuron, blue, that responds to an image indirectly connected to the seed image. The study differentiates between two types of replays: congruent replay, which occurs when the direct neuron fires shortly after the seed neuron, followed by the indirect neuron. This order of firing mirrors the actual order of images in the learned sequence, maintaining the temporal structure. Incongruent replay, which is the control condition, and is when the indirect neuron fires before the direct neuron, which would indicate a reversal or mismatch with the learned sequence. The replay analysis uses data collected during breaks between exposure sessions, referred to as B1 through B7. These breaks provide an opportunity for researchers to observe a spontaneous neuronal activity without any active stimuli being presented. By analyzing these intervals, the study explores whether neurons are reactivating the learned sequence patterns, maintaining the timing relationships between stimuli, even when participants are not actively involved in the task. The diagram shows the expected order of firing for the seed, direct, and indirect neurons during a congruent replay. In their analysis, Fried and his team discovered that neurons in the hippocampus and entorhinal cortex gradually adapted and aligned their activity with the sequence of images on the pyramid graphs. These patterns develop naturally without any direct instructions to the participants. Moreover, the neuronal activity not only reflected the likelihood of upcoming stimuli, but also preserved the encoded patterns even after the task ended. It is clear to see how our understanding of human memory has evolved since the case of Henry Molaison, from understanding the role of the hippocampus itself to Fried and his team providing the first evidence of how the brain uses similar mechanisms to represent what appear to be very different types of information, space and time. These landmark studies have provided the necessary foundation of understanding human memory and have allowed for future research. Thanks for watching. This is a project for Dr. Zhang's neurobiology class at Mizzou, fall 2024.